Okay, so welcome everyone to the next session of this workshop where we are going to see today how we can use the next generation data, sequencing data to do the genome assembly and quality analysis. And for example, we will be taking the microorganisms or the bacterial genome to do the assembly. Okay, so and then we will also check the quality of the assembly, how we can select the particular assembled sequences or the contig sequences. So I will explain it to you a little bit as introduction, and then we will go through the workflow. So in the genome sequencing shotgun approach, okay, where we take the total genomic DNA extracted, we do the sequencing on any of the sequencing platform like Illumina or any other sequencing platform. From there, we receive the short reads. These short reads can be assembled to form the longer sequences. Why? Because our ultimate goal is to get the complete genome sequence. And to do that, first is the high throughput sequencing where we produce enormous amount of sequences, short sequences, because this platform has the limitation, but we have the advantage of getting the high number of or large number of the sequences. So these sequences are assembled to form the longer sequences. They are called the contigs. These contigs can further be assembled into the longer sequences, and they are called the scaffolds. These scaffolds may have the ends, may or may not. So there might be the ends there in between the gaps of the contigs when the scaffolding is performed, or sometimes they are filled properly with the nucleotides. And to perform the scaffolding process, there is the requirement of the paired end sequences. So these paired end sequences, as I have already explained it to you earlier, that when we sequence any template DNA from both the ends, forward and reverse side, they are called the paired end sequences. They have the advantage over in during the assembly process, like assembling the repeat regions and also making the longer sequences from the context like scaffolds. Okay, so once we have the assembly, we should or we want to know the sequencing coverage. Why it is important? Because the sequencing coverage is the number of the reads which are covering a particular reference DNA. The number of the bases of reference DNA is being covered by the number of the reads there, and that is the terms of the, in terms we call it the coverage. So suppose we have a genomic segment here. So how we can identify the coverage of the sequencing? So that is very easy. You can see here n is the number of reads, L is the length of each read divided by the length of the genomic segment, L. Okay, so that will tell us the coverage. For example, we have the 1000 base pair sequence here, reference sequence, that could be genome or anything, template DNA, which we have used for the sequencing. And then, okay, suppose we received 200 sequences or the reads out of the sequencing platform. Then each read has the length of 51. So the coverage of the sequencing data will be 200 into 51. That is the number of reads multiplied by the base pairs divided by the genomic segment or the template DNA, which we reference DNA, which we sequence there. That will tell us the exact coverage of the sequencing data which we have obtained. In the terms of Illumina sequencing data which we receive, most of the times when we do the complete genome, a whole genome sequencing or complete genome sequencing, we prefer to get up to 100x coverage, which will give us the good assembly. More than that is also expected, but at least 100x coverage is a very good amount of data to start with. Okay, so how we do the assembly process? There are various assembler available. So most of them, they are based on the K-mer based assembly, and also the algorithm what they follow is the De Bruijn graph. So earlier there were assemblers like Nubular and other assemblers. They had different algorithm. What they were doing, they were just taking the, suppose this is the initial segment or the reference DNA or genomic DNA we have sequenced here, and the short reads were generated. So the alignment was being performed, and then the consensus sequence was created there. But the problem with this is that they are very computational resource intensive. They require a lot of computational resources. So to avoid that, there is the benefit of using the K-mer based process that requires less memory compared to the direct alignment of the reads. Okay, so in the K-mer process, K-mer based assembly process, what is done? The reads have all the possible K-mers. The algorithm is designed to select all the possible K-mers, like there are tetramers, pentamer, hexamer, like A, C, T, C. So suppose in this case here in this example, 4 mers are being taken here as the K-mer length here. So the all possible K-mers will be taken out like A, C, T, C, and then C, T, C, G, and then T, C, G, A. Likewise, it will be taken throughout the whole all the reads, and then the repeated K-mers will be removed from this data, and then they are aligned using the K minus 1 alignment there of each of the K-mers. So a graph is created, and then the consensus sequence is created here. As I have showed you here in the 4 mers K-mer value, it is not taken in the real life or real assembly process. Why? Because if we take the even number of the K-mers like 4, 6, 8, 12, 14, then they will form the palindromes in the sequences, and then that will confuse the assembler, the software or the script which we are running for the assembly process, and the assembly will be not good, or there will be erroneous assembly outputs. To avoid that, we select the odd number of the K-mers instead of the even number. That is 3, 5, 7, 9, like that. Okay, is it clear?
Yes, sir.
Okay, so what we are going to do here, that is the genome assembly workflow, and then the downstream analysis. There are various steps we have followed, like we receive the raw FASTQ data, suppose, from the sequencing platform. So there are various tools available like FastQC, PrintSeq, NGS toolkit, Tribomatic, cut adapt, adapter removal. You can use any of these tools and then perform the quality filtering and get the high quality FASTQ data, FASTQ reads there. Once we have the FASTQ reads here, okay, so what we can do, we can perform the assembly of these short reads as I showed you earlier in the previous slide. Then assemble genome in the form of the context scaffolds, or even we can create a circular genome if we have a good amount of data with us, short read data. This assembled genome can be used for the annotation process, like using the PROCA, RAST, CAS, PGAP, CARD, and anti-smash datasets. The other way we can also follow here, using the high quality FASTQ reads, you can do the alignment of the short reads to the reference genome, any known genome you want to study. Okay. Please give me 1 minute. Okay. And we can do the variant calling and SNP's prediction there to see the differences between the any known pathogen and then the isolate which you have conducted. You can do the same analysis with any of the reference genomes with your own test organisms genome. Okay. So there are various assemblers available as I showed you here, like SPAdes, Velvet, CLC. This is the paid software, CLC Workbench. So Lenovo is also a free software available. IDBA, Kanu, MetaHit. So there are various assemblers available for the assembly of short reads data. Okay. So here we can see the Velvet assembler, which is the oldest one. Like it was, you can say, developed earlier during the NGS data analysis. Velvet uses the deep blue graph based on the K-mers to assemble the short reads, and it works in 2 modules, that is Velvet H and Velvet G. First, it creates the hashes of the data, and then Velvet G uses those hashes and creates the de Bruijn graph based on the K-mers, and then forms the contigs. There is the advancement, and then another group has developed the SPAdes assembler. This assembler, you run only one command there, and then it will perform the assembly of the contigs. And if there is the you can say paired and data is available, then it will also perform the scaffolding of those sequences. So this SPAdes also works with the de Bruijn graph, and we have to define the k-mers, or we can use the default k-mer values. It will select the k-mers based on the read length. So I will show you how you can use this later on. So it works in the different steps in the assembly process. Error correction is the first step. So it does use the HMM KHMM method for the correction of the reads. If there are errors available or present in the data, sequencing data, it will try to correct that. After that, it creates the de Bruijn graph, repeat resolution is performed, and then finally contigs is obtained there. Okay, so there are the various steps in the SPAdes, but we need to run only one command, and then we get the assembled sequences. After that, so how you can download the SPAdes, I will show you through the website and also show you the commands, but you can install the SPAdes there using sudo apt install SPAdes. Okay, like this command, as I showed you here somewhere. No, so I will tell you that command. You can install the SPAdes using sudo apt install in Ubuntu, or you can also download the binary version of the SPAdes tool from the particular link. So I will show you how you can download the SPAdes software. Once the assembly is performed, you can check the quality of the assembly, quality of the assembled sequences using Quast software. So there are various other softwares also available. One of them is the Quast. What it does use the reference genome alignment and also the quality of the assembly. Like it will tell you how much length we have received for each of the sequences. And if you want to compare, we will also get the comparative data there, that which assembly is good. Suppose you can use various different assemblers, and then you can employ the Quast tool for the assessment of the assembled sequences quality. So you can install the Quast using conda install and then -g bioconda Quast inside the Anaconda. You can also install it from this particular link which is given here. So let me show you. If you have questions, you can ask, and then I'll go to the website link for the SPAdes and Quast.
Sir, I have one question.
Yes.
The number of contigs that are generated by different assemblers will be different. So how we can choose which one is the best one?
Okay, yeah. So that all the stats is based on N50 value. You must have heard about the N50, L50, and there is also N75, N95. Those values are available there. So I'll explain it to you in the quality assessment.
Okay, thank you so much.
Okay. So here is the official website of the SPAdes software. You can download the various help documents like manual here.
Sir,
You can also
your
check that.
screen is not visible.
Oh, I shared it, but it's, yeah. Now it must be visible. Is it?
Sir, it's still
No,
not
still not
It's
visible.
not. Okay, let me share it again. Is it visible now?
Yes,
Yeah.
sir.
Sir, now it's yes.
Yes.
Thank
Okay,
you.
so here you can see the official website of the SPAdes tool. Here you will find all the documentations of regarding the SPAdes, like description, manual, subscribe. So you can subscribe for the their newsletter using the email you can provide there, and then they will send you regular emails there for updates regarding the SPAdes assembler. So you can download here, download SPAdes binaries for the Linux, SPAdes binaries for Mac OS, and source code also you can download. Suppose you downloaded this version of the Linux and it didn't work on your system. So what you can do, you can download the SPAdes source code and try to compile it by providing all the requirements, like all the dependencies. If you face any problem during the use of this binary, I told you the other way you can use to download the SPAdes is just type the sudo apt install and SPAdes. It will download the version which is available in the repository for you, the latest version. It might not be the 3.5154. So what you can do, you just download this, right click and save link as to a particular location in your system. Okay. Then you will find the manual also for the SPAdes here. This is the SPAdes. If you want to do the Quast thing, so you can go to the Quast website here, and also you can go to the source force. So this is available from the GitHub and the source force both. Okay. To install the Quast, they recommend you can install it from the source if you want to. Otherwise, you can also follow the Conda installation method. So you need to install the Anaconda, and then you can run the Conda install minus the Bioconda and then the Quast tool. Okay, so is it clear how you can go to and get the SPAdes and the Quast tool here? So we will
Excuse
move
me,
back.
sir.
Yes.
Are the SPAdes is specific for the Linux system, or is it also in Conda also we can use that? Like Quast is for both Linux and this Conda,
Yes.
but for SPAdes, is it Linux specific?
Yes, yes. You can also install, download it and install inside the Conda environment as well. Okay.
Okay.
Yeah.
Okay.
So it is available from three sources. Either from the you can say Ubuntu repository, Debian, and
Okay.
then you can also download it from the binaries. You can download directly from the GitHub, and the third one is the Conda installation. So you can also use inside the Conda environment.
Okay, sir.
Okay.
Okay. Thank you.
I think the terminal is visible to you.
Yes, sir.
Okay. So we have the SPAdes. As I showed you, I downloaded the version of the SPAdes 3154, the latest version here in this current directory. You can download it anywhere. Only thing, if you want to run it, because this is the binary, you can check the SPAdes directory, which I downloaded just now. So you will see here bin, that is the binary folder which contains all the binary scripts inside to run the SPAdes. If you go further inside the bin ls, you will see all the scripts which are required to run the SPAdes. They are present here. Okay, it does use the H-hammer tool for the error correction of the data or the short reads. Okay, so other thing I have done, I have installed the sudo apt install SPAdes, and it did install the SPAdes in the system location, but the problem was there that it was the older version. If I show you that SPAdes.py -v, which is installed from the sudo apt, then you will see the older version is installed there. So if you want the recent version, you can just download the binary and give the path to run the SPAdes. So how you can run? If you have installed it using the sudo app, then you just write the spades.py and minus minus help. It will show you all the help available, options available for the spades to run. Okay, but we want to use the latest version of the spades, so we can use inside from inside this directory. You can add this to the path so that you just write the spades and it will run for you. Otherwise, you can do here current directory spades, give the path, and then inside the bin there is the spades.py. It will run again. Okay, so this is the options for the spades 3.15.4, right? And this was the SPAdes 3.13.1. Okay, so you can download the different versions also. Suppose you have run and you publish the data or you send for the review. So suppose they are asking for some more modifications, so you can use the same version of the assembler for the analysis, because most of the times the data or the outputs, they get varied when we use the different version of the softwares. So that is the one limitation of the using the different version of the software. So we need to be sure which version we have used during the analysis. Okay, so we have here data inside this directory also. These are the reads which we have obtained from the Genomics. If you remember, we created the forward paired, reverse paired, forward unpaired, and reverse unpaired. So we will use all this data for the assembly process. Okay, so let's check again the help option for the SPAdes. Okay, to run the SPAdes, we need to provide the output directory name where all the output will be stored. The results will be stored. Isolate is for the, if we have suppose multiple multi-cell data, then we can use the isolate option here. That this is coming from the isolate, because we can also run the SPAdes for the single cell single cell genome assembly. That is the data which is coming from the multiple displacement amplification. You can also assemble the metagenomic data set here. Biosynthetic SPAdes mode you can also run here. Corona SPAdes you can also run here. So these are the new other options which has been added over the time in the SPAdes tool. You can use here the RNA-seq data assembly also inside the SPAdes. Plasmid assembly you can also use here for the using the SPAdes, and then use this plasmid option here. Metaviral SPAdes pipeline for virus detection. So it will help you to detect the viruses present in any of the data set. Metaplasmid, it will also help for the in the metagenomic data set to find out the plasmids there. So these are the newer options which were added. These options were not available in the 3.13.4 version. If you see here, these options are not available in that. So these options are available in the recent version of the SPAdes. Okay, so the data can be given here as input as the interlaced forward and reverse paired and reads. So what is interlaced is like forward read and then the reverse read for that pair. Then the forward read and reverse read, they kept into the single file. Okay, but we can also provide the -1 and -2. So forward read separately and the reverse read also as the second read. Then single line data also we can provide here using -s option, file with the unpaired reads. There is also merged option. Suppose we have merged the forward and reverse reads there. So we can use that file by providing the flag merged and then the file name. Okay. There is the option of paired and 1, 2. So you can use the paired and library numbers here and then give the data input there. Different libraries you can. Mate pair libraries can also be used. So mate pair libraries are what? They are, yes?
Sir, can we use BWA for alignment for a genomic DNA? That
Yes. Yes.
matlab, instead of मतलब SPAdes or something like this online database, मतलब online type of tool, मतलब we can
Yes, sure. You can use. There is a portal available, K-base.
that is up to us. Okay.
Yes, so there is the K-base available. I think it is free to use, and you can use most of the tools available there. You need to create an account, and then the data is kept there for 90 days. So you can use these tools over on that portal, on that web server. Okay. Galaxy also you can use if you want to. Okay, so there is the options of these, like the make pair reads also can be provided here. High quality mate pair library number. So if you have the different libraries there, you can provide the library numbers there as the input. Sanger sequences, like the sequences which we obtain from the capillary sequencing output. So we can also use those sequences as a file name along with the short reads. We can also use the PacBio reads assembly here. Nanopore reads also can be used for the assembly. Trusted and untrusted context files, file with the trusted context, file with the untrusted context, they can be provided there. Suppose you have some sequences you are doing the some assembly and then curation there, then you can provide these sequences as the find out, like they are the trusted and untrusted sequences, we want to separate them. Then the pipeline option is you can only run the error correction. You can correct the reads and don't run the assembler. So you can use these options here, like various options. Only assembler can be run without the error correction. Suppose you have the error corrected data, or you have used some other tool for the error correction. You don't want to use the SPAdes and then the hammer tool to run the error correction. So you can run the only assembler mode here without doing the error correction. Careful mode is the tries to reduce the number of mismatches and short indents. So it will take some time, more extra time, but it will be much more authentic assembly. Checkpoints, so you can run the checkpoints, last checkpoints there, save intermediate checkpoints, last all, and then continue is the continue run from the last available checkpoint, and then restart from. So you can also run it from certain parameters. You change the new parameters and then run the SPAdes there, right? The most important thing is the memory here. Memory is, by default, it uses 250 GBs of RAM if it is available in the system. But if you have more memory available, suppose you are doing the metagenomic assembly, then you can use the memory option to the 500 GB or something higher. You can also provide the number of threads. Default is 16 or the number which is available in the system, or you can also provide -T to give the multi-threading, more threads than 16.
Also,
Okay.
sir.
Yes.
Sir, actually you said Galaxy to use, but I am not able to register in that Galaxy website.
Pardon? You are not?
Able to register in the Galaxy account.
Why?
Sir, it is not accessible. I think so. I'm not getting the link in that
I
tool.
haven't used it for a long time, so I'm not sure. You can try the K-base then. Okay.
Okay, I will try
Yeah.
that. Thanks.
Okay, so let's run the assembly. I have run one assembly here. That is the defined one. So what is the defined? Let me show you both the commands. This is the defined assembly which I ran. So I explained to you that was K-mer based assemblers, or the K-mer and then the De Bruijn graph based assemblers. So this is the SPAdes. We are using the isolate option if we are running the recent version of the SPAdes. If we are not running it, you will get the error if you are using the older version of the SPAdes. So careful when you are running the 3154, then you will only be able to use the isolate. Otherwise, you just remove this option for the older version. Okay. Then there is the -1, that is the forward read, that will be the forward paired dot fast queue, and then the -2 is the reverse read, reverse paired dot fast queue. Then -S is for the single end read, which doesn't have the proper pairs there. So that is the forward unpaired and reverse unpaired read. Then output is the assembly space defined, the directory where we want to create all the outputs and the result file. So you can rename here. As I run it, so we will run it at the default K-mers. We will not define the K-mers here. I provided the -t number of threads we want to use here. And then -k option is for the K-mers or K-mer values we want to use. So there is the different K-mer values we can define here. We can give the SPAdes. So it can start from the 21 or much lower, but that is not required because the read length is still longer, and that will also take some more time. And if we go higher or longer K-mers, that gives us the much better assembly. And then what SPAdes does here, it uses all the information of different K-mer assemblies and then give us the final output. So you can define here. I haven't selected here for the particular K-mers. I just wrote the random numbers here for the odd numbers like 33, 45, 57, 65 till 127, because SPAdes supports only the K-mer length which is up to 127 bases long. It doesn't support longer than 127 bases. So if you run this, it will run all of these steps here, 33, 45, one by one, and then finally 127, and then at the end, it will give us the output. So I'm going to remove this because I already ran this one using the -k option. We will run the defined K-mer. There is the problem with the name of the files here. Forward pair and breaking. Okay, and then unpaired. Name of the files was written wrong. That was the problem. I just corrected it. I will also change it inside there. So I will press enter to run this, and it should work. Okay, I'm running wrong. SPAdes. We have received the error because I'm using the wrong version. So I'm going to run the recent version from the path where we have downloaded it. SPAdes and then bin spades.py. Okay. So what it has done here, it has selected these number of the K-mers 21, 33, 55, 77, 99, and 127. Okay, and what we did, we used the different K-mers starting from 33 to 127. So much more steps we used there. So this is also good. You can use the defined ones, but you can also try the different combination of the K-mers and then do the assembly and then check the quality of those assembly sequences. Okay. Do you have any queries?
Sir, I have one.
Yes, please.
I don't know whether it will fit into this or not, but at times when we are performing assembly of bacteria, let's say, and with Illumina short reads, then there are certain gaps because
Okay.
the reads are short. So is there any recipe that we can fill those gaps?
Yes, that is most important thing we want to do actually. So the first thing you can do is go for the nanopore or the PacBio sequencing. Okay, to fill the
Let's say
gaps.
we have financial crunch, we can't perform both nano or pack bio. We have only funds for Illumina only.
No, it won't cost you too much, I think. So you can look into the prices and is it a prokaryotic genome?
Yes, a bacterial.
Yeah. So you just look for the codes, and I think it will help you. It won't be much expensive. So the thing is here. Other thing you can do is designing the primers and then go for the gaps, but
That will
that
be
will
very
cost you
lengthy.
much time. Yes, that will cost you a lot of time.
So
Okay.
the only thing is that we have to perform both long reads as well as short reads for a better depth, and then perform assembly combining all those reads.
Nowadays, this is the best approach, I would say.
Okay,
Yeah.
thank you.
Okay, so this is finished now. We have the default assembly. We did not provide the number of K-mers there, which we want to use. Then we also have the defined K-mer assembly. Let's go inside this defined one, 3D. We have here all the K-mers which we selected, 30, starting from 33 to the 127. Now you will see here the SPAdes log. So in future, suppose you want to check the different parameters, or you want to change some parameters, you can always check the SPAdes log here, which parameter you have used to run the SPAdes, right? And then also all the output. Let's see here. The most important output here, what we want to see is that contigs.fasta and the scaffolds.fasta here. Here. Okay. If we check the scaffolds first, it has the number of sequences written here, node 1, length of the sequence, and then the coverage of the sequence. It is calculated by the SPAdes. Then there will be the next sequence, node 2, length, coverage. Till the end we have 21 sequences here. Okay, 21 sequences in the defined K-mer, where we have selected different number of the K-mers. Okay, we have total 21 sequences. If we go back to the default assembly of the SPAdes, so you will see here the drastic difference in the same tool, but with change in few parameters only. So if we go here inside, and if you check the scaffolds.fasta file, okay, sequences are different, like length is different, and also the coverage, right? If we go at the end of the file, we will see there are 108 sequences available. The length is 81. We don't need that because we cannot use the sequences which are smaller than 200 base pairs. Even though if we select the sequences which are longer than 200, we still get more sequences in the default assembly here. Longer
Sir,
than, yes,
I have
yes,
another question. Like generally, this number of scaffolds will be less than context.
yes, it should be.
But yes, but in few data we've seen that the number of scaffold is more than context. So how is that possible?
Um, from the NCBI data, or it's your data?
No, in one of our lab, mate has performed this genome assembly, and the data
Using
she received,
using using which tool?
I think SPAdes only. So the data that she received had in one of the set, the number of scaffolds were more than number of contacts. So it was surprising because ideally number of contacts should be more and scaffolds should be less.
Yes, I haven't seen this kind of output, so I'm not sure.
Okay, sir. Thank you.
There might be some problem or errors or some. I think, yeah, there must be some problem with the data. Okay, so we have here 46 sequences longer than 200 base pair, but we had lesser number of the sequences in the defined K-mer. So now we have seen, like, okay, till now number of sequences are telling that we have the best assembly getting from the defined K-mer output. This is here. This is the defined one. Okay, but is it really right? Is it really correct? So what we can do, we can check the quality of the assembly using the Quast tool. So we have a genome also, that is the Mycoplasma pneumoniae strain 11. So this is the closest, you can say, you can say that Mycoplasma pneumoniae, because this is also from another strain of the Mycoplasma. So I have downloaded it to compare the assembly, but we need to be sure that in the microorganism there is a lot of genome rearrangements and other kinds of horizontal gene transfer steps are being always being performed or always being done. So in that case, it is not a wise idea to compare with the reference genome. However, if you are doing some kind of resequencing analysis and checking for the mutations or genomic rearrangement in the same strain, then this is a lot helpful for you. Okay, but however, I want to show you that we can use the reference genome here. So let's check the Quast here. I have installed it inside the Anaconda using the command anaconda install
Oh, excuse me, sir.
-c. Yes.
Sir, is there any way to learn more about this SPAdes system?
About what?
This is
SPAdes.
SPAdes one. Yes, sir.
SPAdes. You can go through the manual which is available from the SPAdes website,
Okay,
and
sir.
you can also go through the literature, the papers from the developers they have published. Okay, Bioconda and then Quast. So you can install the version which is available from the Anaconda, download it there. Once it is installed, you can start using the Quast. There is various options available for the Quast. Like, we can provide the output directory to store the results there. Then Reference genome we can provide, as I told you, if you are doing some kind of comparative analysis of the previous assemblies, you can use the reference genome to compare the recent assembly. Okay, you can also provide the features or the genomic features there, the ORFs and then their annotations there, using the GFF bed and CPI or text files as the input. Minimum contig length you can provide here, as we can submit to the NCPI as the 200 nucleotides long sequences we can submit it here. So we can use here 200 instead of default, which is 500, which is much longer. So we can change this parameter. Number of threads you can use to parallelize the process, and then there is more other options available, like if you are using the eukaryotic assembly quality analysis, fungal, large genomes you are using here, so you can provide these flags to tell that this is particular genome. Okay, you can use all these K-mer stats, K-mer size based analysis you can define here. So based on the different parameters you can use here for the analysis. Minimum alignment, so the minimum alignment length to the genome here, that is default 65. So you can change this alignment region also, or the length there. So there are various options available. You can explore all these options depending on your requirement, like extensive miss size. So lower threshold for extensive misassembly size. You can change this. Like default is 1000, so you can change this parameter. Any of the parameters you can change here. But depending on that, you are knowing which parameter stands for what. If you are changing like okay strict NA, sorry, this one ambiguity score. So if it is like 0.8 and 1, it should be between this one, and default is 0.99. If you are reducing this ambiguity score, you must be aware that what you are going to see in the output. Otherwise, don't change any of the parameters until you are sure. Okay, so we can run the quast here. I will copy the files, scaffold files. I will copy from the assembly outputs. So we scaffolds dot fast, and I will give the name here, scaffolds define dot fast fa. Not, this is not the define, this is the default. So scaffolds dot fa, it will be the same. And then I will write here cp assembly define scaffolds faster to scaffolds define dot fa. Now we have both the scaffolds here. You can get all of your assemblies using the different assembler or different tools. How many assemblies you have, you can import all of them here to run the quast. So as we have two here, we have used this based on two different parameters, defined K-mer and default K-mer values. We can run the quast here, minus o quast out, and then we will give here minus r is the reference genome name genome file, that is this one, and then the sequences. You can use all those options if you want to. Okay, I haven't used the option here minus 200 for the minimum length. That was the problem. I forgot to do that. Okay, so it is finished. Elapsed time this much, 45 seconds, and board notices 0 warnings, non-fatal errors 0. So it was passed, it was completed. We can go inside the output directory, quast out. We see here there are output files and directories which were created. They contain the report as HTML, PDF, text, TSV, TXT. Different format of the report output is available there. So we can check the HTML file here from this directory. Open it any of the web browser. I will open it. Okay, let me share this. Okay, now it is shared. So this is the output here from the Quast. Okay, is it visible to you?
Yes, sir.
Yes,
Okay,
sir.
nice. So we have here genome statistics. For the scaffolds, I didn't give any name, so it was the scaffolds defined. And this is the this was the scaffold default, and this was the scaffolds defined. If we check here the genome fraction, so you see worst is denoted by the red in the color color as red, and the blue is as the best, and the median is showing here as the white. So if you see here, the scaffolds defined the K-mer values which we define here. The genome fraction is covering more to the reference genome with respect to the reference genome. That is 98.4%. And when we use the default K-mer values, we did not provide, it was 96% compared to the reference genome here. Duplication ratio is 1.001. So it is more or less similar, but you see only 0.001% difference is there to the other one. Largest alignment to the reference genome is 238248 base pairs, and here 200,000, 21,985, right? So this one has the longer alignment compared to the defined one. Total aligned length is higher for the defined K-mer assembly. NGA50 is the length of the sequence, okay, which covers the 50% or more of the reference genome or self the assembly. Here, in terms of the NGA50 is with respect to the reference genome, which we have used here. So NGA50 means, suppose we have the sequences of 300,000, 200,000, and 112,000. Okay, 112,000 sequence length or base pairs are there. So if we combine 300,000, 200,000, and 112,000 base pairs together, they will cover the 50% or more of the reference genome. Right? Is it clear to you? NGA 50? If it means that the sequences are longer than 112,000, and when we combine this one with the longer sequences, they will cover the 50% of the genome. And here we can see this is better here for the defined one, not for the default one. Okay, is it clear? NGA50. The same way LGA50 is telling us that the number of the sequences which are covering the 50% or more of the reference genome. So it is same for the both assemblies. It is not different. Then again we can come here for the misassemblies. So here it says that we have more misassemblies in the defined K-mer, not in the default one, which has lesser. Like 2 misassemblies are there. Here we have 3 misassemblies. Again, to consider back to the reference genome, whether it is the closest or most closest one, or it is somewhere far from this one. So the misassemblies are based on the reference genome. So we need to consider that also. There is, we can also extend the report, right? So this is based on the genome alignment to the reference genome. Unaligned sequences here, partially unaligned length. These are the sequences which were unaligned. Mismatches are there also, like to the reference genome. So we are not going to consider this because we are not sure. We just randomly selected. But if you are doing some re-sequencing analysis type of study, then that is most important thing you need to consider. Statistics without reference. So now this was based on the reference genome output. Then we can also check the output without the reference genome. And if we go here, the largest config is here. Total length, it is covering more. Total length in more than 0 base pairs, 1000 base pairs is also higher here. Total length more than 5000. So overall, we see here without the reference genome, the quality which tells us that this one is much better, the defined one. So what we can do, we can select this one for the further analysis. And there are few plots also, they were generated, like the cumulative length of the assembly here. So you can see this one has the shorter assemble sequences or shorter assembly than the defined one here. The red one. Yes?
This has been defined by the tool.
No. This is defined by us.
Okay.
This was the K-mers. We use the two different parameters here. We defined the K-mer and we did not define the K-mer.
Okay, fine.
Yeah. So without the reference genome, this one tells that this is a better assembly output. Okay. And then you see here the NX, the number of sequences which when you add to the bucket, how much it is covering here with respect to the assemblies. NAX is here, also the alignment of the sequences there. Then NGX is based on the genome alignment, which we use here for the as the reference genome alignment. Okay. Then the NGAX is there, also the alignment of the to the reference genome, how it is covering there. Misassemblies, where they lie in the assembled data. So this has the 2 misassemblies here, and then the one here, the 3 misassemblies here, 2, and then 3 here. One misassembly in the default one, and then the second one is here in this region. Okay. GC content, if you compare, this is more or less the same which we analyze the data when we analyze the data using the fast QC for the short reads. That was in the range of 41% here, right? So this is the sum of the statistic outputs, and based on this you can compare various different assemblies or tools, and then you can select the best assembly for your downstream analysis. So in this case we are going to select the defined K-mer here, defined K-mer based assembly for the annotation process, which we will be doing tomorrow. Okay, so if you have queries you can ask. I will try to answer, and then we can close the session.
Excuse me, sir. Sir, was there a class on Saturday?
On Saturday, yeah.
No, there was no class on Saturday.
It was on Friday, I think.
No, sir. Saturday I was not able to connect, so that's why I
No,
asked.
Saturday there was no class. Saturday, Sunday, no class. We got an email.
Yeah, we got
Okay,
an
okay,
email
okay.
about
Okay,
that.
it was on 23rd.
Okay.
So, sir, are you going to teach us about that RNA-seq? Because Saturday it was about that RNA sequencing.
Maybe